Hello, for this new episode of my publication at a glance, I'm very happy to welcome today a duo, Rémi Châtre, who is currently a postdoc, and Estelle Blochouse, who is a PhD student at the University of Poitiers working at the Institut de Chimie des Milieux et Matériaux de Poitiers. Thank you for your participation.
Thank you, Guillaume. Today we will present the work of our latest publication. There are two major challenges in cancer chemotherapy: the diagnosis of the disease and the efficacy of the treatments. Within this framework, we envision a new strategy called induced volatile-omics for the design of tumor-activated therapy. The tumor microenvironment is known to have several upregulated enzymes, such as glycosidase, that are targetable with product of anti-cancer agent. In order to find the best target enzyme, we have prepared a cocktail of probes sensitive to different glycosidase that can release a specific volatile organic compound for each enzyme. After the administration of the cocktail to a tumor-bearing mice, its metabolization within the tumor releases the volatile organic compound, which can be trapped and analyzed. Then, the upregulated enzyme is identified. We can synthesize a product sensitive to that specific enzyme and finally evaluate its anti-tumor efficacy.
Here are the 4 probes that we work with, each targeting different glycosidases. We can use those probes with 2 different principles: in vitro, on blood cells or tissues, and in vivo on animals. Both have their advantages. First, we use the probes on tissues to know which enzyme can be a good target for a disease. We compared healthy tissue, represented by kidneys, and cancerous tissue from breast cancer. We mix the probe with the tissue in airtight conditions, and after a few minutes, we can collect the volatile molecule with the solid phase microextraction fiber and analyze it by GC-MS/MS. For the first set of experiments, we obtained 1 enzyme with no significant difference, 2 targets that were significantly higher in tumorous tissue but needed a high concentration of probe, and the last probe was used in a low quantity and was giving a high significant difference between the healthy tissue and the tumorous one. Then, the second type of experiment was done in vivo, because in a whole organism, some results are not transposable due to the interaction between organs and cells of the full body. The probes were injected to healthy and tumor-bearing mice, and after 30 minutes, we collected and analyzed the volatile organic compounds. Luckily, we obtained the same result with no significant difference for the 2 first probes, a small difference for the third one, and a high significant difference for the last targeted enzyme, telling us that this enzyme can be a potential target for the development of a selective chemotherapy.
Following the results obtained with the VOC-based probes, we decided to synthesize a new monomethyl auristatin E product sensitive to the enzyme that is upregulated in the tumor microenvironment. Indeed, when the product accumulates in the tumor, the activity of the N-acetylglucosaminidase hydrolyzes its glycosidic bond, leading to a cascade 1,6 elimination followed by a decarboxylation, resulting in the release of the active monomethyl auristatin E in tumor. When we evaluate its biological activity in vitro over 2 cell lines, we incubate the cells with the product in absence or presence of an excess of the enzyme. The results indicate that the product is well substrate of the enzyme because the IC50 are the same compared to the free monomethyl auristatin E. We next evaluate the therapeutic efficacy of the product in vivo for the treatment of mouse bearing orthotopic breast cancer. The results indicate that the administration of the product every 2 weeks leads to a dramatic reduction in tumor volume compared to the control group, without inducing any side effect. As a result, 100% survival is obtained for the group that is treated with the product. And finally, at the end of the experiment, 4 mice were cured of the disease.
With this new technology of induced volatomics, we were able to screen the activity of several tumor-associated enzymes, which led to the identification of a target. This target was used to design a new anti-cancer drug that have a selective delivery. This new drug finally showed an impressive antitumorous activity in mice. This cocktail of volatile organic compound-based probes can be also used for the exploration of enzymatic dysregulation linked to other diseases, such as viral infections. Thank you for your attention.
Thank
Bye.
you.